Good morning, everyone. Welcome to the 2025 ICTEV meeting. Happy to join you here, and always happy to start off the Heart Valve Society meeting. I'm Josh Hutchison. I helped co-organize this meeting, but as most of the hard work was done by Davy Vandervalk from Leiden and Eindhoven, so I'm going to invite her up here to say a few words to get us started.
Perfect. Good morning, everyone. Very nice to see all of you here. Some familiar faces, some new, fresh faces. Welcome to the International Conference of Tissue Engineered Heart Valves 2025 in Cairo. So I want to start off the day by thanking our main sponsor, which is the Material Driven Regeneration Program based in Eindhoven. Eindhoven, Utrecht, and Maastricht in the Netherlands, and these universities work together to make new smart materials for tissue engineering of organs, among which heart valves. So it's a very fitting sponsor for today. So to introduce today's session, I wanted to relate it a little bit to the place where we are here in beautiful Egypt in Cairo. Yesterday we went to visit the pyramids, and I couldn't help but think about the relation between the pyramids and heart valve tissue engineering. So these pyramids were built, the three that you see over here, by King Khufu and his son. This is the second one, and his grandson is the third one. And they were made 2600 BC, so they've been there for centuries. Which made me think that's kind of what we want to do. We want to build structures that will be there for centuries. If that doesn't work, maybe just for the lifetime of one patient. It should be okay. How can we do that? Make structures that will last for centuries? I think we can only do it by looking at the relation between the structure that you want to make and its function. So this relates really nicely to our first session, where we will talk about development and structure-function relationships within heart valve tissue engineering, and relate that to native valves. So we have another few really nice sessions. In the second session, we will talk about addressing the translational barriers and challenges of heart valve tissue engineering around the world. In the third session, we will talk about cell matrix interactions, decellularization and recellularization of heart valves and scaffolds, and the host response. And in our last session, we talk about a new era of heart valve tissue engineering, and we talk about new technologies that can help us move forward with the field to make these pyramids. We have a lot of nice invited lectures today. I'm really excited to hear from all of the invited speakers. But we're also very excited to hear from our young talents, and therefore we also have a Young Talent Award at the end of the day for the best presentation. So I want to do some shameless self-promotion and also introduce the Young Professionals team here at HVS. So we'll be going around this whole week, and we have some nice activities for the young people in the crowd. We have a breakfast event on Saturday, a social event on Friday night, and a meet and mentor session where you can meet established people within the fields and talk to them about how to make your own career move forward. So if you want to join our Slack, please follow the link or the QR code. So I want to thank all the people that helped organize today. So the team of this year, Dr. Carlijn Bouten, Dr. Jesper Jortnaas, and Dr. Joshua Hutchison, thank them all for helping us organize this. All the moderators that are helping today, all the presenters that are presenting today, also the HVS organization team, specifically Katie, Missy, and Jessica, thanks for their work. The team here based in Cairo, the technical team, and specifically also Dr. Ismail El-Namamsi and the HVS 2025 for having this beautiful location here in Cairo. So without further ado, let's move to the first session.
right. Thanks, Davey. So again, I'm Josh Hutchison. Before we start the first session, just have a couple of brief reminders and announcements here. So if you have a question at the end of each session, or at the end of each talk, please wait for the floating microphone that I've told will go around. Also have a notice here for the Pearl Room. The Pearl Room is located on the ground floor past the garden, and been told to look out for the peacocks there. If you're not yet a member of HVS, we invite you to join and get involved. Please visit the registration desk in the Canyon pre-function area for more information. This year, the HVS Education Fund was created with the goal of supporting the next generation of leaders in various disciplines of heart valve disease. Please consider donating. You can visit the registration desk to learn more. Should you need a certificate of attendance for your return home, please also visit the registration desk. And please be sure to visit the electronic display posters in the foyer, and they can also be viewed from within the mobile app. And I also invite you to please join us this evening from 5 to 6:30 p.m. for the HVS and ICTEV welcome reception. So I hope to see you there. So I'll let my co-moderator introduce this first session.
Hi, I'm Dr. Rachel Cahalan from the University of Galway. So I'm really happy to open our first session today. So it's entitled Native Valve Blueprints, Development, Structure, Function, Relationships, and Heart Valve Tissue Engineering. So we're going to start this morning with an invited lecture from Dr. Josh Hutchison, and he's going to tell us all about native valve cell heterogeneity and associated implications in valve development, maintenance, and disease. Take it away, Josh.
Thank you so much. So I'm excited to be here to give this talk this morning, and generally at meetings I give an overview of the heart valve, the aortic valve, and I'll skip this here, but I am going to be focused on the aortic valve today. I kept this slide up here just so I could not forget to recognize Dr. Daniel Chaparro, who just defended his PhD dissertation 3 weeks ago. Is also part of our young professional network here at HVS, and many of these slides were ripped off directly from him. He has some super high resolution images in some of these, which are the computer's having some problems with this morning, so I'll also blame him for that as I give him credit, but we'll deal with that as it comes. So we know that aortic valve function is really dictated by this very complex and well-aligned extracellular matrix, this circumferentially aligned collagen fibers, radially aligned elastic fibers that give the valve its necessary properties to stretch and seal the valve during diastole, but not prolapse into the ventricle. And classically, this extracellular matrix is maintained by populations of valve endothelial cells, somewhat similar to vascular endothelial cells, though we've known for the last couple of decades that they do have some distinct characteristics, and valve interstitial cells that are within the interstitium of the leaflets and make the new matrix, degrade old matrix, and allow this valve to function. And then they can also deposit excess collagen and calcification, calcific mineral that leads to valve stiffening. So this is a very classical model. This slide was actually from my PhD dissertation over a decade ago now. But what we've come to realize over the past few years is it's not so simple. We can't think of the valve as this uniform population of valve endothelial cells and valve interstitial cells that are fibroblastic. So on the left here, we have work done from Catherine Yutzie's lab. On the right, recent paper from Joy Lincoln's lab. These are both single cell-based analyses where we can appreciate the diversity of cells within the valve. So even what they're calling interstitial cells here, see if I can figure out how to use this. There, even in interstitial cells, you have a wide variety of types of cell populations there. They're not all just uniform fibroblasts. And even that's not the complete picture. So this is work that we had done with Elena Akawa when I was a postdoc in Boston a few years ago now, that some of these populations aren't even captured in these single cell analyses, likely owing to their difficulty in getting them from the valve. So we know that there's neuronal cells, there's glial cells in the valve that look classically glial and express GFAP, which is a classical glial marker. And we have no idea what these cells are doing. This is work from Adrian Chester when he was working with Sir Magda Yacoub a couple of decades almost ago now, looking at neurons in the aortic valve and putting electrical stimulation across the leaflets. And when they electrically stimulated the valve leaflets relaxed. And when they inhibited neuronal nitric oxide synthase, the valve contracted, suggesting some active regulation of valve mechanics that still is not well understood today. This is beautiful work from back then. We've spent quite a bit of time over the last few years focusing on melanocytic populations of the valve that are still very underappreciated. So we've used mice of varying pigmentation levels, so hyperpigmented mice, hypopigmented mice, and we've shown using some nice structural imaging. This is using 2-photon-based imaging of elastic fibers in red, collagen fibers in green here. This is what a normal mouse looks like. You can appreciate the pigment that we see in these C57 black 6 mice. Our hyperpigmented mice have expanded elastic fiber deposition. Again, things that we still don't quite understand yet. Our albino mice, or in this case KIT deficient mice, have no elastic fibers that we can detect in the aortic valve leaflet, suggesting these cells are very important in patterning. And we can also see that on the gene expression level. We've also noted that there's some asymmetry in these melanocytes that are in the valve. Okay? So if we look at our C57 black 6 mice, this is work done by Daniel Chaparro, just kind of keeping note of things. He noted that 70% of the time in our wild-type mice, we see pigmentation only in the right and non-coronary cusps, not the left, whereas 30% of the time, they're in all three, and he's quantified that here in both males and female mice. We've now done almost 200 mice, and this penetrance remains. So that's interesting because of the known penetrance of bicuspid aortic valves in eNOS deficient mice. It's the exact same penetrance, this 30% penetrance of bicuspid aortic valve malformations in eNOS deficient mice. And of course, this is the most common congenital heart defect, affects 1 to 2% of the population, but accounts for 50% of all stenosis-related interventions at this point. So what Danny did was he looked at these mice and said, well, if we see the same penetrance where we have 70% of the time these melanocytes, no pigment in those wild-type left coronary cusps, but 30% of the time we have pigment all three, well, let's look at some eNOS mice and see if that same penetrance matches the penetrance of the bicuspid valve malformations. And sure enough, it does. Every time we see a bicuspid aortic valve in an eNOS deficient mouse, it's pigmented across the board. I will state, however, this doesn't seem completely sufficient to drive bicuspid valve malformations. We see some tricuspid valves that do have pigmentation in all three leaflets in these eNOS deficient mice. But to characterize, this is brand new, by the way. This is work that the last mice that we've done so far were just completed last week, so we're still very underpowered, but I just wanted to show you this. Just again, driving home these atypical cellular populations in aortic valve morphogenesis and patterning. What we've done is we've gone back and crossed eNOS deficient mice with a black 6 albino mouse. So this mouse has a deficiency in tyrosinase and can't make pigment to produce black 6 albino eNOS deficient mice. Okay. This is what those valves look like. You can appreciate there's no pigmentation in the black 6 albino eNOS mice. And we're now quantifying the penetrance of this bicuspid valve malformation. Very underpowered and not significant yet. But so far, our penetrance is approximately 10% in the eNOS deficient mice compared to the normal 30% that we see. So the eNOS albino mice compared to the normal eNOS deficient mice. So we're still working on this, but again, some evidence that these cells are involved in some kind of remodeling. We've been working closely with Joy Lincoln recently. I mentioned her study here. So this is using single nuclear RNA sequencing, looking at the heterogeneity, again, of all these different cell types and how they cluster here according to differences in gene expression. And she recently sent us all the data from her melanocyte population, and these are the differentially expressed genes. If you look at the genes that are significantly differentially expressed only in that melanocyte group compared to the other groups, and run a gene ontology analysis, all of these categories were found through gene ontology. So not surprisingly, melanin synthesis was there, but then regulation of TGF-beta signaling, tricuspid valve morphogenesis, mitral valve morphogenesis, cardiac morphogenesis, ventricular septation, all of these genes known to be associated with cardiac formation, with septation. So I think that this is just some demonstration that it's not just fibroblasts likely, going again back to Dr. Chester's work, all the way through what we're seeing in melanocytes. There's other cell types there that are controlling valve function and valve remodeling. And valve disease itself is likely a mixture of genetic predispositions, as I was talking about with that bicuspid valve malformation, perhaps improper remodeling, or improper patterning of these cells in development, and environmental risk factors. So I want to talk a bit about the disease side of things. I've talked more about the development for a moment. And one of our favorite ways of inducing disease is by inducing chronic kidney disease. And there's a strong correlation between chronic kidney disease and aortic stenosis in human patients as well. And of course, the survival outcomes of patients with chronic kidney disease is significantly impacted by the presence of aortic stenosis. So we use a mouse model. This is a high adenine, high phosphate fed mouse. And we're now working on some nephrectomy-based mice as well, where we, again, induce renal dysfunction and show that after approximately 12 weeks, we can get significant calcification in aortic valve leaflets. The calcification's in red here, nuclear staining by DAPI, or by Hoechst in this case, is in blue. This has allowed us to test remodeling of the aortic valve in some of these altered mouse populations that we have. And I want to talk about one thing that we've noticed recently, and this is consistent with human-based data. So these are two clinical observational studies where we know that the non-coronary cusp of the aortic valve exhibits a higher predisposition, a higher burden of calcification compared to the left and right coronary cusp. And this has been known for really a couple of decades now. It's been reported several different times. We see this in our mouse model. So we see in our mice this higher prevalence of calcification in the non-coronary cusp compared to the left and the right. So we're recapitulating the human pathology quite well in these mice, at least from that standpoint. So we found that pretty interesting. This is work that was published just last year, I believe, in Circulation, an AHA consensus statement to talk about all the different knowledge gaps that we have in aortic valve disease. And one thing they identified was this reason of why the non-coronary cusp calcifies maybe before or is accelerated or higher burden than the left and the right. What makes that cusp different? And this is the classical explanation for that. This was put out by Dr. Catherine Otto 2 decades ago now, that the shear stress is likely the explanation. You have differences in shear across these leaflets because of the presence of the coronary cusp during diastole. As blood flows back and refills the coronary arteries, you get a nice high shear, steady flow across those cusps, whereas the one without a coronary artery exhibits disturbed flow. And we know from the vasculature that regions of plaque development and even calcification in CKD occurs in these disturbed flow regions. So it's reasonable to believe the same thing's going on in the valve. But we actually had a little bit of an opportunity here that was unexpected. Again, this is for any trainees in the audience. I'll mention that Danny, my PhD student, did a really nice job of tracking so many characteristics about these mice. And what he noticed was in the mice, oftentimes, the left coronary artery exists where it's supposed to be, below the sinotubular junction, the left coronary orifice, whereas the right coronary orifice generally exists above the sinotubular junction of these mice. So it's higher than what you see in humans. And he quantified this and showed, indeed, this is significant. So we see a range. Each dot here represents where the orifice would have been, the ostium. A range of heights from down below the sinotubular junction, behind the leaflet, all the way up high into the ascending aorta. So in this case, the mouse does not equal the human. Right? So in humans, for the most part, the right and coronary ostia are below the sinotubular junction. But it provides us an opportunity, right? So we can revisit this hypothesis, because when that right coronary ostium gets high enough, the shear stress on those leaflets is going to mirror the non-coronary cusp. This is where my slides are going to get wonky here in a second, so I'll do an interpretive dance if we have to. So here was our hypothesis. Again, the asymmetric onset of remodeling across these leaflets is due to differences in hemodynamics. More disturbed flow, more calcification. More steady shear, less calcification. So we could test that. We worked with a very brilliant researcher, Dr. Asad Mirza, in our department, who does a lot of computational fluid dynamics. And so we can model, based upon where this right coronary ostia is, what the influence is on shear across these leaflets. If you could see this, what you would note is that when the right coronary ostium is very low, we see shear of about 18 dynes per centimeter squared across the right coronary, which matches the left. And there's no sense in going through this too carefully, but when we increase the right coronary ostium really high, that shear drops to 0.34 dynes per centimeter squared, which matches the non-coronary cusp. And so then we plotted right ostium height versus calcification, and it was flat. We see no correlation between that. Again, if the hypothesis this was purely hemodynamics driven, you should see a nice correlation here. So, again, I think this is the last slide that doesn't work. We'll see. These leaflets are different, right? And they're different in ways that we know from development, but don't know well into adulthood. So meaning the neural crest, the endocardial cushion, endocardial cells, and second heart field populate these leaflets, but do so differently, asymmetrically. Okay? It's known that the neural crest has a higher burden, I believe, in the left and right. This is where I can use my slides, compared to the non, and the second heart field is more predominant in the non compared to the left and right. So even from birth, we know that these tissues are different. So we went to do some spatial transcriptomics to try to understand better the differences across these three leaflets. And so if you're not familiar with this technique, we section our tissues. So you can see this would be a blown up of the aortic valve, but you can see the leaflet still here, 3. And we can then track transcriptomic changes spatially and understand which genes are more expressed in the right, say, versus the non and left, and so on. Again, these are very, very new data. But just doing some dimensionality reduction clustering here, we can see a variety of clusters from these, and this is what's important. So if we look at male and female clustering, they're not so different. So at least there's no major differences by sex in these mice. But interestingly, the valve, the aortic valve, clusters distinctly from myocardial tissue, which you would expect. It's a unique tissue. So we see the valve clustering up here. What gave us a little bit of confidence, again, in these early results that we're doing things right, is if we look at differences between our male and female samples, the top differentially expressed gene is a gene involved in X chromosome silencing in females, so that's good. So we see that difference between males and females. And the top gene in the valve versus the rest of the tissue is periostin, which has become one of the preferred Cree drivers of inducing gene expression in the valve itself. If we then just focus on the valve, we don't see unique clustering from the 3 different cusps. However, if we start looking a little more closely at differences, say, between the left versus the right and non, the non versus the right and left, and the right versus the non and left, some interesting things pop out. So first, we see all of these phenotypic markers, DCT, PMEL, TRP1, downregulated in the left. That is associated with melanocytes. You remember I told you that oftentimes that tissue is not pigmented. So again, consistent with our phenotypic findings. So we see that again here. And also, we see things like myosin heavy chain 11 differentially expressed across these 3 leaflets, indicative of smooth muscle cells or perhaps myofibroblast activation. But interesting, one of the genes that we see come up is PALM-D, upregulated in the non versus the left and right. And if you've been keeping up the literature, the aortic valve, you know, PALM-D is one of these genes associated with aortic stenosis. It seems to be upregulated in the left, in the non versus left and right. Still really early. We actually just got some more sequencing done last night, and this seems to be consistent. So we're still working on this. So is it truly due to shear stress why the non calcifies before the left and the right? And we don't think that's the complete story. We think there's some influence between environmental factors, say like CKD, say like hyperlipidemia, mechanical-driven factors, so that shear likely plays a role, but also perhaps genetic factors in terms of how these cells infiltrate the valve tissue during development. Right? So it's this kind of complex interplay, and I think that what we need to be doing moving forward is better connect what we see up here, these differences in these developmental lineages, to these cells in the adult tissue that are not fibroblasts. These melanocytic cells, we have shown there's similar cells in humans. These neurons, these glial cells, what are they doing during disease, and how does that connect back to genetic predisposition? And so what I really want to leave you with is, do we need to consider this heterogeneity in tissue engineering? Right? So we often look at cell infiltration later on in tissue engineering and say, okay, we have momentum positive cells in the interstitium. We have an endothelium. But no one ever looks and say, are we recapitulating any of these cell phenotypes that might be critical for valve function? Is that important? I don't know, right? But I think we should at least be looking for them because these cells are there, and all indications are they're playing a role in development and perhaps in maintenance of things like valve tonality, thinking about the neurons, even into adulthood. So it might be something worth considering as we move forward and start moving more toward implementation of our strategies. So with that, I would certainly like to thank all the folks in my lab, all my collaborators, and funding. Thank you.
So I believe we have some microphones that are going to come around if anyone has questions for Josh. Yeah, there's one here at the front.
Very nice talk, Josh. Zishan. Obviously a little self-serving, but I start thinking about, pulmonary valve versus aortic valve. One has coronary sinuses, the other one doesn't. Your comments on, have you looked into it, differentiations, and all of this, I think it really highlighted the complexity of heart valve structure and function. So I appreciate that, but your comments on it.
On the pulmonary valve itself and
Yeah. Versus the aortic differentiation?
We haven't looked a ton yet. At least in the melanocyte story, what I can tell you we see is that any time that left coronary cusp is pigmented, the pulmonary is also pigmented. What that means, we don't know yet. We also do know that some of the genes that we're focused on now that are driving both pigmentation and elastogenesis are also implicated clinically in patent ductus arteriosus. So again, some involvement in that septation between the pulmonic and systemic circulation. So we think there's something there we haven't checked yet. Yeah.
Hello, see you again. So question regarding to pigmentation. So what's your thought regarding to the fundamental cost?
My thought regarding what now?
Yeah.
Regarding the pigmentation and
Yes.
the purpose of it? Or?
Yes.
Yeah. Yeah, we don't know. So that's been one of our biggest holdups is that we don't see pigmentation in human leaflets, right? Now we've gone back and looked, and we did a lot of this work with Elena, and we see cells that express dopa-chromatopteromerase, PMEL, so all these melanocytic markers. So we think that there's a similar population there. Now why in the mice they also produce pigment, we don't know. But there seems to be something linking perhaps, so we've looked at this elastin phenotype, right? Elastin, tropoelastin itself is trafficked through these acidic vesicles inside the cell that are actually very similar to melanosomes. So perhaps in mice, the same cells kind of have that same machinery doing both purposes, whereas in the humans, for some reason, it has separated. But we're still actively investigating that.
Another question regarding to your work associated with the clinical outcome. You know the structural heart disease is very fundamental, the cause for life management. Regarding to your work, can you give some suggestions regarding to your work associated with the clinical outcome?
With clinical outcomes?
Mm-hmm. Thank you.
Sure, we think so. I mean, in terms of, especially the bicuspid valve malformation story, right? We know that that's highly linked to prevalence of aortic stenosis, and then downstream poor ventricular function. And so we think that understanding the ways in which these leaflets are structurally patterned is going to lead us better to understanding of the development of aortic stenosis, again, which is directly linked to clinical outcomes.
Josh, just quick question. Thank you so much. As always, beautiful work. Thank you for your progress and everything. That's great. So I was just wondering, when we look at the black 6 mice, so it's naturally that the melanocytes produce this melanin, right? But when you look at the albino mice, they probably have the same cells,
Right.
but they just not labeled naturally.
Right.
So I'm just wondering if you're missing them somehow.
Yeah, I think so. Well, I don't think so. I know so. I know so that we've looked in albino, we've looked in BALB/C mice, so all these mice that don't have like colored coats, and they typically don't have pigment in their valve. The cells are still there. the cells are still there. And in fact, in the mice, they always express tyrosinase. They always express all of the full panel of melanocytic genes. Again, in the humans, we're missing a couple of those genes involved in melanin production, but the same, again, similar cells there in the humans as well. So I think there is some kind of atypical melanocytic population that's there.
Thank you.
Okay, if there are no more questions, then I think we will move on. Thank you, Josh. Okay. So our next speaker is going to be Patricia Caruso, and she is going to tell us about structure function characterization of fresh human heart valves.
Okay. Good morning, everyone. Okay, that's okay. Thank you. I'm Patrizia Caruso. I'm from Palermo, and I'm recently achieved my PhD. So I'm going to show you the results of my project. And thanks to the Fondazione Rimed, that is I work here as specialist. So my project is about structure and function characterization of fresh heart valves. As you know, the tissue engineering heart valves has tried to achieve the most challenging cardiovascular disease in medical field. So in the last years, the most heart valves have been mechanical and bioprosthetic, but these devices have some limitations. For example, the mechanical valves offer the advantage of durability, but they require a long therapy anticoagulation, and they to prevent the risk of thrombosis. On the other hand, the bioprosthetic valves do not necessitate ongoing anticoagulation treatment, but their long-term performance remains plagued by failure modes and leaflet tears. So the advances in heart valve, the tissue engineering heart valves, have tried to find a new solution and using biopolymeric valves, using, for example, scaffold or decellularized matrix tissue to increase and to achieve new technologies that can substitute the mechanical and bioprosthetic valves. Also in this field, we have seen that the heart valves have been characterized using animal models. For example, porcine valves that are used for cardiovascular devices for their anatomically similitude with the human heart valves. And also the ovine valves have been used for biomaterials because for the tissue regeneration and to validate some devices and cells growth. So the purpose of this research is to give a deep analysis of the fresh human heart valves into four main aims: the structural analysis at the macroscopic level and microscopic level, and a function analysis in plane mechanics and out-of-plane mechanics. First of all, the fresh human heart valves that we used were from the Ismet Hospital in Palermo and also from the Core Foundation in Pittsburgh. The analyses were performed within 24 hours from the explantation. This project is also part of the ERC Biomatter project with the principal investigator, Dr. Antonio D'Amora. So first of all, the structural analysis want to give an idea of the morphological geometry at the microscopic level. How we did? We obtained the valves and we dissected the leaflets and we measured the thickness using a micrometer in 15 points spanned from the free edge to the belly region and the commissural zone. As you can see, the mitral valve presents the higher values of thickness compared to the other valves. We wanted to correlate these values also with the transvalvular force. We used the ImageJ software to calculate the area of the leaflets, and we found in literature the transvalvular pressure with the higher values for the mitral compared to the other valves, and we found the transvalvular force. As you can see, there is consistency with the values of the thickness. To complete the analysis, we used also a MATLAB code to obtain the maps of the heterogeneity of the thickness for each leaflet. And as you can see, there is higher differences between all the leaflets for all the 4 valves. After that, we wanted to compare the fresh human samples in terms of thickness measurements with the other 2 species, the porcine and ovine. As you can see, there is a statistical differences in particular for the aortic and tricuspid valves when we compare the species. In particular, the ovine present the lower values of thickness when compared with the other 2 species. To complete also this analysis, we used the same MATLAB code to have an idea of the maps per each valves, given the idea of the heterogeneity of the thickness in all the area of the leaflets. To have also a deep characterization of the structure of the leaflets, we used the 4 main staining techniques, such as the hematoxylin and the eosin valves, the Mason trichrome to underline the presence of the collagen, and also the picrocyrus red, and at the end the Alchan blue to underline the presence of glycosaminoglycans. Because, as you can know, the atrioventricular valves, mitral and tricuspid, have a different layers composition compared to the semilunar valves, the aortic and pulmonary, and we want to see the differences between all the leaflets. To complete the analysis, we wanted to quantify also the presence of the collagen using the Mason trichrome staining technique, and we used a MATLAB code to quantify the percentage of collagen per each valve. And as you can see, the left side valves, the mitral and the aortic, present higher values of collagen in compared with the tricuspid and aortic, and this is consistencies also with the thickness values that we found. For the structural analysis at the microscopic level, we with this analysis, we want to have a deep idea of the extracellular matrix that component each leaflet. Why to do this? Because some parameters, such as the fiber bundle collagen diameter or the orientation index of the extracellular matrix, is not well reported for the fresh human samples. So first of all, we just reviewed the literature with established protocol, and we develop our own protocol that is using enzymatic such as the trypsin for 3 minutes, and then other detergents to just remove the endothelial cells without disrupt the structure, because we want intact the structure. And as you can see, after the decellularization of 48 hours, the structure of the leaflets is intact, and also this is confirmed by the ACM analysis. And also, to be sure that we removed all the cells, we performed also an histological analysis where the cells are removed and the structure is intact. So to have the parameters that we want to characterize, such as the orientation index of the fibers, we use the MATLAB code based on the Chaduri-Carlo algorithm to have an idea of the anisotropic structure of the leaflets. As you can see, all the valves present an anisotropic structure higher of the values of 0.5, that is the isotropic structure parameter for us. And we also found the fiber angle distribution for all the valves. After that, we use the ImageJ software to have an idea and collect this parameter never reported for fresh human samples, that is the fiber bundles diameter. As you can see in this case, the atrioventricular valves, mitral and tricuspid present higher values of fiber bundles diameter when compared with the semilunar valves. To complete this analysis, we compared the values of the fiber bundles diameter with the other two species, finding also in this case that the ovine species samples present the lowest values of fiber bundles diameter when compared with the porcine and human samples. So we completed the structural analysis. We can go with the functional analysis. In this case, we wanted to see which is the response of the valves when they are subjected to a biaxial loading test. In this case, we harvested the samples, a square sample of 10 per 10 millimeter from the belly region. That give us more idea of the mechanical response of the leaflets. Distinguish two main direction, the radial and the circumferential direction. We used the Lagrangian equistress control protocol with a maximum of 400 kilopascals. As you can see, the mitral and tricuspid present a highest values of stiffness in the radial direction when compared with the semilunar valves. This can be related with the different coaptation system because the atrioventricular valves, the coaptation system is due to the presence of the chordae tendina. Instead, for the semilunar valves, they are more compliant in the radial direction and present highest values of anisotropy between the two directions. We distinguish the different valves to have an idea that all the valves present the highest values are stiffer in the circumferential direction, and this can be correlated with the presence of the collagen fibers that are oriented in this direction. To complete the functional analysis, we used a custom-made bending device to have more information about the bending rigidity of the leaflets. This is a custom-made device where we harvest the tissue from the belly region, also in this case, oriented in the radial direction. We used 4 polypropylene wire to obtain our markers, and the test was performed in both the negative and positive curvature with a maximum curvature of 0.12 millimeter. We also distinguished the two main surfaces of the leaflets that called against curvature, that is subjected to the highest values of pressure during the cardiac cycle, and that called with curvature. As you can see, in this case, the against curvature presents the highest values of rigidity when compared with that with curvature, and also in this case the atrioventricular valves, the mitral and tricuspid, shows highest rigidity compared with the aortic and pulmonary valves. So in conclusion, a point of strength of this work is that we used the fresh human samples and analyses were performed in 24 hours. We correlated the thickness measurements with the transfer force, giving a first step of the analysis and comparison between structure and function of the valves. As we see with the SEM analysis, the leaflets have an homogeneous anisotropic structure, and the human valves showed higher values of collagen fiber bundles when compared to the other species. With the mechanical test, we can say that there is greater stiffness in the circumferential direction than in the radial direction, and the atrioventricular valves displayed greater bending rigidity when compared with the semilunar valves. For future developments, of course, the structural characterization can be increased with some other parameters, such as the porosity measured in the human tissue of the extracellular matrix. We can integrate our biomechanical data into computational models to optimize the prosthetic valves designs. And the study of the cellular proteome could contribute to elucidate the molecular mechanism behind the cardiac valve and disease, and this can be a true key that connects the structure and the function of the valves. Thank you for your attention.
Do we have any questions?
Thank you.
Thank you for a very nice overview. Maybe a future prospective question. So many groups have analyzed biomechanical properties of valves and their structure function properties. If you want to add this to computational models that can predict how tissue develops, should you then also not include the mechanical properties of developing valves, so younger valves, growing valves? What's your opinion on that?
Thank you for your question. Yes, I think that this is just a first step. As you see, the fresh human samples were very few. So increased analysis can give more response also for the mechanics because we have few samples. But for future, one of the analyses that we want to do is, of course, the proteomic analysis and see which is the differences between the leaflets and all the species, of course. Thank you.
I have one quick one, just curious.
Yes.
From the data, it looked like your pulmonary valve might have been the stiffest valve in the circumferential direction. Is that correct? And what are your thoughts on that and maybe implications for things like the Ross procedure, for example?
Come again?
And implications in terms of like Ross procedure when we, put the pulmonary valve on the aortic valve side.
You mean this?
Yeah, exactly.
Yeah, first of all, there are few leaflets, so we have to increase this number. As you can see in the circumferential direction, there is such as compression of the tissue. This can be correlated with the, I show you, I have this situation. As you can see, if we increase in this soft tissue the stress that we are applying, it seems that the collagen fibers manage their orientation when we increase the stress. So instead to have stretch, we have a compression of the tissue. But I think that it's better to increase this number to see if something that is can be replicated or is affected with the few leaflets that we analyze it.
I think we have one quick one.
It's a quick question regarding technical question. So for your desterilisation, before you treated by detergent SDS at 2100, you also treat by pepsin for 30 minutes. Mm-hmm. What's the concentration for pepsin? Thank you.
Thank you. Thank you for your question. It was 0.1 milliliter of trypsin because as we saw, we tried to apply different protocols. And we saw that if we use too much trypsin, the structure was disrupted. And this is what was something that we didn't want, because we want intact the structure to have only the extra matrix and to remove all the endothelial cells and have all the fibers well out of the tissue to be analyzed. This is because we use only few means of trypsin. Thank you.
Let's thank our speaker again. So we'll move on to our next talk, which will be given by Dr. Cassandra Clift, who will talk to us about cross-species proteomics applied to explanted cardiovascular biomaterials, enables distinction of donor versus host extracellular matrix profiling in bioprosthetic implants.
It's a long name. probably should have shortened. Okay, thank you so much for the introduction and also the opportunity to talk to you all today. And it's fitting that we're starting to talk about better ways to characterize both the native valves, and I'm going to be talking about a characterization technique that focuses on bioprosthetic valves, specifically really relevant to xenogenic materials. So I'm a postdoctoral research fellow in the lab of Dr. Elena Aikawa, and I don't think to this audience I really need to describe the problem that we're facing here, just to say that we focus on calcific aortic valve disease, which, as you all know, there's no pharmacotherapeutic interventions that currently exist. So at the Center for Interdisciplinary Cardiovascular Sciences here at Harvard Brigham and Women's Hospital, we're really focused on drug target discovery and biomarker discovery, which kind of leans more towards the protein level. So when I'm talking about characterization going forward, especially in the context of pharmacotherapeutics, I'm going to be focused in proteins. And then just in the context of this disease, why we care about that. So as you know, healthy aortic valves, they become calcified, they fail. And then a major mediator for therapies now is bioprosthetic valves. And then how we're focusing on that in the field is primarily with glutaraldehyde fixed tissue from other species. And in the instances of this study, we're going to be focused on those made with bovine pericardium. So the pericardium, if you don't know, is this matrix-rich tissue on the outside of the cardiovascular, or on the outside of the heart, that's glutaraldehyde fixed and then put into the shape of a valve and implanted into the patient. But those, unfortunately, also have a propensity for failure. So just like a native valve, they will also calcify and fail over some time. And then right now, the processes governing bioprosthetic valve degeneration are insufficiently explored and not really well understood. So when another researcher, just sitting here to my left, Rachel, came to work with Elena Aikawa in our group, she was really interested for her grant proposal focusing on the key proteins that are implicated in bioprosthetic valve degeneration. She's actually going to be talking in Heart Valve Society later this week on this specific topic, looking into the biology. And then she came to us because she wanted to use proteomics, right? She's interested in the protein level. So she was assigned to work with me because my background is in mass spectrometry and in proteomics. And I said, Rachel, I love this. This is really important to the field. However, I can anticipate some technical challenges that we need to solve before we really get into the biology. And part of that, because we are on the protein level, we need to consider this kind of cross-species contamination problem. And it doesn't need to be contamination, is kind of what I was thinking. It could be an opportunity to look into biology of these two different species. So there's actually about 78% overlap between the bovine and the Homo sapiens that we need to consider, and this is not in the context of proteomics, just in the context of what we know of all of these orthologous groups between these species. So, I then changed the objectives of the study, not just to utilize a pipeline that we will develop to profile the ECM, but first we need to develop this novel processing pipeline first to delineate between these two cross-species peptides. So bear with me a little bit on this project. It's going to be kind of going into mass spec method development and some quantification theory, but I hope I can walk you through it so you can appreciate kind of the advantages of working with a mass spectrometer collaborator as opposed to kind of submitting samples to a core facility, especially as proteomics becomes more and more attainable, and it's something that a lot of us are doing. So just to kind of go into proteomics, when you submit samples to a core, normally what happens, you give them a protein prep. Sometimes you even just give them tissue or your cell culture sample. So traditionally proteomics is done in the bottom-up approach, where you take the protein and then you digest it, usually with trypsin, into peptides. You use chromatography to sort primarily, and then it goes into the mass spectrometer. Those peptides become fragmented so that we can directly sequence their amino acid sequences. And how that's done is that, in a mass spectrometer, it just sorts things by mass to charge. So you get a mass spectra, and that eventually needs to be annotated into your peptides of interest. So again, those peptides of interest in the cases of bioprosthetic cross-species are going to be coming from bovine and human, and that's something that we need to consider when we're doing our tagging and the database searching. So again, peptide fragments, we need to consider that peptide sequence identification. So just to go over that one more time, peptides go into the mass spec, and then you have something called MS1 or MS2. So you hear tandem mass spectrometry, means you repeat the mass spec portion twice, MS. So for MS1, you have selected precursor ions. These are our intact peptides, and then those intact peptides are selected and then put forward into the mass spec again to be fragmented so that we can directly sequence the amino acid sequence. And that's how we identify mass to charge in that spectra to our peptide sequences. And ideally, when we fragment our peptides, we'll get these breaks really nicely across every single amino acid in the sequence. But unfortunately, that's not actually what really happens when you have your B and your Y ions, your fragment ions. You're not getting all potential B and Ys. You actually get, very few of those. And then why this is important is because if you're really delineating, for example, between orthologous peptides, in this case between bovine and human, potentially that only differs by one amino acid. So you need to make sure that you're really capturing that B or Y ion that is identifying that one amino acid shift. So in the cases of proteomics, usually if you've gotten these workflows before, you're sorting by 2 or more unique peptides. Sometimes 2 unique peptides isn't really enough, especially for cross-species quantification. Okay, so when I teach proteomics, I usually like to bring this up, talking about the proteomics central dogma, which is all about assigning that spectra to protein quantification. So I bring this up because a lot of people understand this biology central dogma, that you work from DNA to RNA, eventually you get protein, right? So proteomics has a really similar central dogma, such that you're starting with peptide spectral matches, so you're working with your raw spectra. Those PSMs are then annotated to peptides, and then that is further annotated into proteins, which is usually the matrix that you'll get when you're working with a core facility. You get some abundance value associated with a protein, but that actually can work its way all the way to the PSM match. So that's a really simplified version of the proteomics central dogma. In reality, it's actually quite complicated. There's many different steps of feature detection. You also need to consider where they're, I haven't talked much about chromatography, but where they elute off the chromatography column matters with all this feature detection. So feature detection just means there's so many different factors that we need to consider when we're kind of eventually going downstream for quantification. So most proteomics that we do is usually label-free quantification, and this is done on the MS1 level. So again, that's the precursor and not the fragment ion. However, like I mentioned, when you're wanting to look between bovine and human features, what if they're only distinguishable by their fragment? So basically what I'm saying here, this is all a long prelude to say we need to change our quantification strategy if we're doing cross-species proteomic. We can't use a traditional triptych pipeline that most core facilities will be using. So how did we approach this problem? Essentially what I'm doing here is I'm combining an intensity and count-based proteomic approach. So most people will just do intensity-based, but I'm actually going all the way back to the peptide spectral matches, so back to the first step in the proteomic central dogma. And essentially what this allows us to do is more robustly quantify these cross-species peptides. So now we not only have a database that we're searching against, so all known amino acid sequences against human and bovine, we're putting that in there, but we get more accurate feature mapping and more accurate peptide to protein tagging. Okay, so now that we've identified this novel pipeline, let's try and apply it to some biology and see what we find. Okay, so starting off with this really wonderful cohort that Rachel brought into Elena's lab, we have lots of different tissues. There's a whole separate project she'll talk about where we're comparing this to the native, but today I'm just going to focus on the bioprosthetic valves. And then here we're segmenting this into different disease segments. So we have our non-implanted control, which is really important because that's essentially our background database. And then the valves kind of will degenerate in various ways. Sometimes they don't degenerate at all. We can tag these as non-degenerated. They look very similar to the non-implanted control. And then they also followed up with histopathology with Elena's expertise. Sometimes we also see areas of thrombus, neotissue formation, which is defined as new human deposition. So essentially cells will come in from circulation and then deposit brand new ECM. So we see that, as well as calcified regions. So again, first part of this quantification strategy is to create that custom limited background file that we can search against because we want to limit our permutations of what we're searching. So I don't want to search against every single potential bovine protein, only the ones that we know that are going to be potential contaminations. So we include this in our search. We put the non-implanted controls, and this, which is actually just kind of like step one of this study, is the first time that anybody has used omics to really exhaustively profile what is in a bioprosthetic valve that is used. So that's another aspect of this study. But as you can see, it's a lot of extracellular matrix proteins. That's not unexpected there. So ECM glycoproteins, as well as structural collagens, non-structural collagens, as well as some ECM regulators. Okay, so now that we have our bovine background, we can kind of do some overlap here in terms of how much is shared with the human potential that we have coming into the valve, and about 98% of those genes are shared. And then again, we're doing proteomics, so I want to consider just the tryptic peptides because we're digesting with trypsin to do bottom up, and of almost 20% of all tryptic peptides actually have this potential to be mistagged between species. So consider that if 20% of all of your data could potentially be misquantified. That's actually a lot of potential room for data misinterpretation. So now that we're accurately tagging our peptides to the right proteins, there's a couple different ways that we can kind of annotate these proteins. One, bovine specific. We never found that human sequence there. We only ever found confidently the bovine sequence with that bovine specific amino acid. Two is orthologs. So we actually found both the bovine and the human, and they did have a differentiating amino acid between those sequences, and we can kind of delineate that way. Cannot differentiate. So while we did find a peptide, it could have been for bovine or for human. It didn't align with a region of those sequences that had a delineating amino acid. And then again, a human specific. So while there is a delineating amino acid in the bovine, we actually didn't find it. So as you can see, just between quantifying the shared peptides between orthologs and those that cannot differentiate, in some cases, we were never ever going to be able to differentiate. I don't care how good your sequencing and quantification is. There's, in sometimes for proteins, almost at 100% sequence overlap. So in those instances, we're not doing cross-species. There's no way to do cross-species. But then part of what we're doing here is also kind of changing how the search algorithm considers the scoring and therefore the confidence by your species. And what we showed here is that, and I won't go into this too much, but essentially when you're doing proteomics, you want to set false discovery rate thresholds, so how confident you are. And then part of the calculation for your FDRs is based off of the length of the peptide, so that we showed that there's no difference between the confidence, even when considering length between bovine and species. So we're confidently tagging both of these proteomes. And then when we look at orthologous species, so those that we can very confidently delineate between bovine and humans, we find a lot of them are extracellular matrix proteins. And Rachel and I were interested in how that ECM is changing in terms of how that bovine is degrading and the new human, or that neotissue is kind of being deposited and leading to fibrosis. So this is great for us. And then what we can do kind of first overview is look at these orthologous profiling over the species. And one way to do this is just with a heat map. We can kind of pick out here. Some instances are blue, so those are going to be low, and then they kind of move into a high abundance protein, for example, looking across this heat map. And I'll leave Rachel a little bit later to talk about different biologies that we're interested in. I'm just going to focus on the method and kind of how you can use this new quantification strategy. So this is one way to visualize your data. But then another way is I wanted to be completely unbiased, and there's thousands of proteins we identified, so I don't want to kind of manually go through a heat map and look through every single one. That might have an interesting shift, right? So I want to unbiasedly look at bovine-specific degradation, specifically in the context of ECM, and human-specific deposition. So in order to do that, we can kind of consider our proteins being tagged as 0 as non-present, for example, so those that cannot be differentiated, human-specific, bovine-specific, human ortholog, or bovine ortholog. And here, 0s and 1s are just representing not present or present, right? And then when you kind of lay this out, you can think of these like vectors. So this is essentially a vector with 5 dimensions, 1 vector per protein. And then what I want to do is kind of calculate unbiasedly the distance between these vectors, because that's going to tell me how dissimilar these protein depositions are. So essentially what we get is a lot of data. Our matrix represents over 1,000 vectors. This is one for each protein. It's in 25 potential dimensions because I've been talking about the species tags, that ortholog versus specificity to each species. However, remember we also have our disease segmentation tag, that like neotissue, thrombus, things like that. So it's a lot of data, which is why it's important to be unbiased. But essentially what we can do is kind of calculate the distance between these vectors and then plot a protein-protein interaction network where the node represents the protein and then the edge weight represents the distance between the vector or the dissimilarity between those two proteins. So then what you can get is a network like this, where this is representing most proteins that are dissimilar across segment, independent of species. We're going to pull out one of our favorite proteins, collagen 1A1, just for a representation of now you can very accurately quantify. Basically, this whole project came from me wanting to make this plot very, very accurately. You need to be able to do a box plot that will count all of the peptides that are very specific to that protein quantification, and then look how that changes across disease segments. So as you can see, for example, collagen 1A1, looking at the neo-tissue region, so this is by histopathology defined as something that has a lot more new tissue deposition. It's unsurprising that we see in that region specifically a statistically significant increase in collagen 1A1. There's a lot more proteins to kind of look at. I'm just showing collagen 1A1 as like the example, our quality control check, right? We expect that to happen. Okay, but then what's nice about this is that I also kind of quantify on the peptide level too. So if you're interested in like specific collagen domains and how those are changing, if you're interested in collagen post-translational modifications, this is a really interesting way to kind of probe into that further. Another thing you can do with this quantification strategy is to investigate, for example, the bioprosthetic implant length over time and change how certain protein quantification change across species due to length of implantation. So how does the degradation and deposition profiles across these proteins change as a function of the length inside of the human? Okay. And then essentially the last thing that we wanted to do was kind of compare this to what most people in the field are doing, which is just searching against the host species as opposed to considering the background proteome, in this case of the bovine. So here we did this comparative study between our cross-species data processing and the human-only data processing. And in terms of identifications alone, you actually don't see that much difference. But I don't actually care about identifications. I care about quantification, because that's where the data misinterpretation and that biopoetry you eventually write when you do your pathway analysis, that's where all of this comes into play, not in IDs, but quantification. So just to kind of quickly give an overview of those plots that I'm about to show, essentially what we did is a rank analysis between the human-only search, so when you're considering the kind of normal quantification method, not our improvement, versus the cross species. If anything's falling on the y equals x line, it means that they are similar. Underneath is underrepresented quantification or versus overestimated quantification. Okay, and as you can see, not a lot of proteins fall on the y equals x line, which means that if you were to do a traditional kind of like core facility search against these, you would be over or underestimating the majority of your proteome. So a lot of misinterpretation for biology is all I'll say there. And then this happens in a disease segment specific fashion. So showing that the under and overestimation and that therefore the quantification changes as a function of your disease segmentation. So just to kind of summarize, we did develop this novel processing pipeline. We're using it in the context of xenografts, but you can also use it in the context of, like, for example, looking at microbiome contamination, like in fecal proteomics, or a lot of people do patient-derived xenografts in the context of cancer. And then we did determine some profiles of some key ECM proteins that are implicated in bioprosthetic valve degeneration. And with that, I'd like to thank our group, and especially Rachel, for coming to us with this really interesting question.
We may only have time for one short question if there's something out there. No?
I scared everybody with mass spec quantification.
I'll ask one quickly.
Sure.
So you've nicely gone between these different regions in terms of calcified and non-calcified. Is there a way that you could ever go back and say more anatomically where you see, like, say, bovine collagen degradation and human deposition across the valve leaflet itself? Is that something that could be done?
Yeah, so we're kind of doing that like macroscopically now just by splitting into segments, but I think a great way to answer that would probably be with like some of these imaging proteomics techniques where you can like spatially map on the peptide level. So like for MALDI, for example, would be really great to answer that. Yeah.
All right, thanks.
Thanks.
All right, we will move along, and our next talk will be given by David Vandervalk, who will present on spatial proteomics characterization in situ tissue-engineered heart valve implants from historical ovine animal studies to elucidate material remodeling.
Yes, so hi again. Today I also wanted to talk about the work we did in collaboration between Carlijns Bouten's group in Eindhoven and Elena Aikawa's group in Boston, where we took these previously studied samples from two different studies that were paraffin embedded, and looked at how can we use them again. how can we dive into the processes that are going on there, and specifically, can we do proteomics on these samples? So in Eindhoven, we're interested in situ tissue engineering. And in this process, we take a resorbable scaffold, that's usually electrospun. We implant that into the patient, which are usually animal models at this point, and there it attracts an immune response. This immune response attracts immune cells that start to break down the scaffold, and simultaneously also attract tissue-forming cells that start to build up new autologous tissue. So if all goes according to plan, you get this new autologous living valve. So we know kind of what is going on within this process from previous studies, but we don't really know exactly what happens within the valves at different stages of development and regeneration within these valves. So that's what we wanted to dive into in this study. So for that, we took 2 previous studies. So both were implanted by the group of Jolanda Kluin, together in collaboration with our facility in Eindhoven, with 2 different materials. So we took the explants from the sheep that we implanted the materials into, and we took both the explants after 1, 6, or 12 months. All these samples were paraffin embedded and sectioned for the study. So in this study, we wanted to see if we could use these paraffin blocks and reanalyze them using proteomics. So in order to do that, I went to Elena's lab, worked together with, among others, Cassandra Clift again with her amazing proteomics database. And we went and made this extensive protocol where we deparaffinized, rehydrated, and dried the samples. Then used laser capture microdissection to look at the specific regions that we were interested in to get our samples. And then again did a very extensive cell lysis, protein purification, and peptide purification process for, in the end, to do proteomics. So using this stepwise approach, we could look at the difference between native and tissue-engineered valves. So we looked at the native explants versus our tissue-engineered valves. We could look at the anatomical regions within these tissue-engineered valves. We could look at regeneration over time, differences between materials, and differences around calcification, which I won't be talking about today. So for the native versus the tissue-engineered valves, for this we took out the explants from the sheep, from the position where we implanted the new valve, so in the pulmonary position. So these were the pulmonary explants from the sheep themselves. And then we also took out from the same sheep the 6 and 12 months regenerated tissue-engineered explants. So these samples were processed for proteomics, and that delivered almost 1,300 proteins, which is quite nice for paraffin-embedded tissue. So we then wanted to look at, can we actually see any differences that we've between these native and tissue-engineered explants? So if we look at distinct proteins only, we see that there's 109 proteins that are only expressed in the native explants. So they are not existing in the tissue-engineered explants. And there's almost 500 proteins that are only expressed in the tissue-engineered explants. So those are proteins that are not existing in our native explants, but only in our tissue-engineered samples. So if we look at the networks that are composed by these proteins, we see that for the native explants, there's a lot of networks related to tissue homeostasis. So cytoskeleton organization, cell growth, cell metabolism, cellular respiration, it's all kind of to make a homeostatic valve. If we look at the tissue-engineered explants, however, we see that there's a lot of processes that are related to immunity. So these tissue-engineered explants are distinct in that there are more proteins that relate to networks that are more related to immunity. And if we zoom in on that, we actually see that there's a lot of networks that are related to the innate immune system. So to remind you, these are explants from 6 and 12 months implanted sheep. But the innate immune response is still very much involved in these explants. So then we wanted to zoom in on these tissue-engineered explants and look at and see if we could look at the different regions within the valve and see if there's a difference of expression within these regions. So for that, we kind of selected the anatomical regions of the valve, so the anastomosis, the base, the mid, and the tip of the valve. And we did laser capture microdissection, where we cut out really specifically these regions within the valve. We then did proteomics on this and extracted a total of 2,200 proteins, which again is quite a nice result for paraffin-embedded tissue. So as a first analysis, we went to look at the overall proteins expressed within these regions. And if we looked at missing values within these regions, we saw that comparative to the other regions, the tip had a significantly higher amount of missing values. So there were more proteins missing within the tip region compared to all the other regions. So one logical explanation we thought for this would be, well, there's less cells in the tip. Because we usually see this, that when we implant the valves, the cells tend to grow, we think, from the anastomosis site into the tip. So there's less cells present in the tip. So we confirmed this with DAPI staining, where we stained, again, all the regions and then quantified it region specifically. And then we saw the inverse of the missing values, meaning that there's indeed less cells in the tip. So the cellularity actually explains the amount of missing proteins within each region. So if we then look at the distinction of these proteins within the regions, and we look at the differences between the samples, we see that in the, I can use this, I think. Yeah. In the tip region, there's very distinct samples, and the other distinct samples are in the mid region of the valve. So the base and the anastomosis sites, they kind of overlap. We can also quantify this by looking at the differentially expressed proteins within the regions. And then if we look at the difference between, for example, the anastomosis sites versus the other regions, we see that there's no difference in expression between the anastomosis and the base. There's 305 proteins differentially different between the anastomosis and the mid, and there's over 1,000 proteins different between the anastomosis and the tip. So in this you see that the further away you are between regions, the more differentially expressed proteins there are. So we also see this if we compare all the regions to each other. So again, the further away the regions are, the more differentially expressed proteins we see. So this is nice, but we also wanted to see what is in these proteins. So if we then compare the anastomosis versus the tip site, we see that in the anastomosis site, there's a lot of proteins that are related to networks, related to cell growth, protein homeostasis, RNA signaling, and a few bits related to innate immunity. So we see still some remodeling response there in the anastomosis site, but we also see quite a homeostatic environment there. If we look at the tip region, however, we see mainly only immunity-related networks and coagulation-related networks. So this is also something we know already from literature, that immunity and coagulation are very important in the early response to tissue-engineered heart valves. But in this tip region, this also remains over time. We also looked at material differences. And for this, we looked at the two different materials, and we do see that there's difference between the two materials, and that this difference increases over time. So at 6 months, we see some difference in expression between the two materials, but at 12 months, there's a lot more distinct proteins. So to conclude, we did a proteomics analysis of paraffin-embedded preserved tissue-engineered heart valves, And we can use this data set to redefine how the early immune response drive late heart valve tissue engineering remodeling. And that cell infiltration and also coagulation, also immunity, are region dependent. So they're anatomically distinct. So thank you all for your attention, and thanks for all the collaborators that have worked on this project.
Questions for Davy? Yes, we have one at the front here.
I just wonder regarding to the innate immune response correlates with your outcome. Is any kind of potential markers which may be key associated between innate immune response?
The markers with
Yeah.
that, yeah.
Yeah.
So there's actually a lot of proteins that correspond to the innate immune response. So we have these networks that are built up of different proteins. We haven't done a deep dive into specific markers yet, so that's still ongoing. But yeah, there's a lot of proteins that are related to the innate immune response. Yeah.
Thank you.
More questions from the audience? No, okay, I maybe have a small one then. Do you know why do you think you see more like circulating cells only going into the tip? Like, do you think it's a purely hemodynamics?
So I think it's not circulating cells only going into the tip, but I think that's the region that they stay in over time. I think the tip is more in the bloodstream, so it's logical that there's more circulation and circulating cells there. But I also think there's a big involvement of that there's the regeneration response within the tip is not as far as within the other regions. So I really think that this regeneration response is driven from the anastomosis site inwards. And therefore, after a while, you still have more immunity in the tip and also coagulation. Yeah.
We have another question here at the front.
In practice study, and I'm wondering about, did you consider about hemodynamics? Did you say study like pressure gradient? And I think artery stenosis more because the reason is become from pressure gradient. So the pressure gradient maybe cause the stenosis processing. So we have do you have some connection to the protein to or maybe higher pressure gradient make the protein more or something more marker? That's
So
my prediction.
to pressure gradients?
Pressure gradient.
Yeah.
Like
Yeah, so that's more into the related to the calcification, you mean, right? Yeah. So in this sample set, we haven't seen a big difference between the different anatomical regions and calcification. So if you would look at different pressure gradient expressions within a valve, we cannot really relate it here. What I do know from a previous systematic review that we wrote is that in literature, generally, the calcification usually starts at the anastomosis site in the base of the leaflet. Yeah.
Okay, and then in the interest of time, we might move on so we can thank our speaker again. So we'll move on to the final talk of today's session. So we're going to hear from Dr. Jane Grande-Allen, who's going to tell us about native heart valve structure and cell behavior across the age spectrum.
All right, thank you, everyone. I'm excited to be here. I'm going to talk about a few studies from my lab that have looked at the effect of age on the structure and cell behavior within heart valves. And I have to, of course, thank my students up front. This is representing a lot of students over many years. So I've got a lot of lab photos, and I also want to point out that there are people here not only from Texas, but also some visitors from around the world. All right. And what's the deal with the pointer? Is it this one? That one? Okay. And it, oh, got it. Thank you. All right. So I tend to think of mechanics as the driving factor that influences a lot of cell behavior. And so with respect to the mitral valve, of course, there's a very complex anatomy that makes the mechanics possible. The microanatomy and the hemodynamics also contribute a lot to this. So in my lab, we tend to look at a lot of different regions of heart valves like has been nicely described today. But we also consider the microstructure, and so try to think about, also describe beautifully how the layers of collagen, elastin, and proteoglycans are involved, and in particular how they contribute to the overall valve function, as well as driving the behavior of the cells, whether they are the interstitial cells in the interior or the endothelial cells or the other different subpopulations that have been described. So in my lab, we have a context of thinking about what are the biomechanics of the valve. So some key features are that, of course, you all know there is a considerable toe region, and then a region of higher stiffness on the stress strain curve, but also just the anisotropic and layered nature of the valve at the fiber level or the tissue level contributes to its function. But then from mechanobiology, we think kind of both ways, reciprocally, that the interstitial cells, for example, can mechanically affect their environment by making a lot of extracellular matrix as well as contracting on the tissue. But then the heterogeneous microenvironment for all of these cells then influences local cell behavior through the ligands that the cells can bind to. And so the normal valves are fascinating. There's also a lot of valve disease that we investigate. In my group, we focus on either myxomatous mitral valve disease or functional mitral regurgitation, mainly investigating the role of extracellular matrix remodeling, particularly glycosaminoglycans, as well as calcific aortic stenosis, and looking at how aortic stenosis resembles and how it is different from atherosclerosis. But we have also looked at a lot of congenital heart defects, and in particular, I am fascinated by how not only do they develop, how these congenital valve defects arise developmentally, but also how their mechanics and extracellular matrix are different from normal valves. So just one of the things that I frequently talk to my students about is this model of the atrioventricular canal defect, where there is, instead of having 4 heart valves, there will just be 1. And so this can't really replicate a lot of the normal cardiac mechanics or separate blood into oxygen versus deoxygenated domains like you would need in a normal heart. And so as a result of this, for example, congenital heart defect, children are going to need a lot of valve replacements over the course of their life growing up. The valves that are implanted in them aren't really designed for children, and the adult valves that could be made smaller for implanting in children don't really work with their kids' lifestyle or blood chemistry. So there's a lot of need for tissue-engineered heart valves for people of all ages for these various diseases. So the driving questions that have motivated a lot of work in my lab are to understand how the cell and extracellular matrix within native valves behave mechanobiologically and how that drives biomechanics, how that's modified in valve disease, and how we can best model these situations in vitro so that we can have inquiry about disease mechanisms. And what I'm going to talk today is about how the effect of age influences these relationships. And then, of course, we'd like for what we learned to be translated to tissue-engineered heart valves, but that's for you. So today what I'm going to cover is a little bit on material behavior of valves across the age spectrum, extracellular matrix composition, and then a little work on cells with interstitial cell mechanobiology and endothelial cells and how they behave homeostatically. So with respect to material behavior, a work that one of my students did many years ago, Libby Stevens, was to look at different regions of the mitral valve and the aortic valve. And in the mitral valve, we typically, with an eye towards the heterogeneity of the tissue that was nicely described today, for the mitral valve, we tend to look at two domains, and this is the, sorry, it's white here. MVAC is the center of the anterior leaflet of the mitral valve, and MVF is the free edge of the mitral valve. And so this is a very collagen-rich area for the MVAC, and the mitral valve free edge is more glycosaminoglycan-rich. So they behave very differently in terms of their mechanics. And we looked at both circumferential and radial behavior and 3 different age groups. And so the age groups were from porcine hearts, 6 weeks, so suckling pig, representing kind of a pediatric age. 6-month-old to 1-year-old is like the typical age that a lot of us who work with porcine hearts get. I call this your average pork chop age, so it's very easy to get this from the slaughterhouses. And then we got some older 6-year-old hearts from the animal vendors. So to only present just a little bit of the data here, we looked at the higher slope area of the stress strain curve, calling this the post-transition stiffness. And we found that whether we were looking at the circumferentially oriented sections or the radial oriented sections, and just the data here are for the aortic valve, mitral valve free edge, and mitral valve anterior leaflet center, we found that there was an increase in stiffness with all age groups, and with all sections that we looked at, whether this was circumferential orientation or radial orientation. And so this is statistically significant everywhere we looked. There we go. We also looked at the extensibility, or like this is the length. This is a strain measurement. That is the length of the toe region, indicated by the little orange dot up in the curve on the top. And so in terms of how extensible the valves are, we found that there was a decrease in extensibility with respect to age in the center of the anterior leaflet and in both sections that we looked at radially. And so the valves are more stiff and less extensible with age. We also just made some observation about the shape of the stress strain curves, or here this is the load displacement curve, and we saw that if we were looking at the circumferential versus radial direction, there was a sharper transition from the toe region to the post-transition region here. We call this, we superimposed a circle on this and called it the radius of transition curvature. So here there would be a smaller, there we go. There we go. A smaller radius of transition curvature or a larger radius of transition curvature. And for those of you who have done a lot of testing of the mechanics and correlated this with the fiber angles, this tends to be a situation where if you have a larger radius of transition curvature, or this general gradual smoother shape change from a pre-transition to a post-transition domain, that corresponds with more of a mixture of collagen fiber angles. And that is one of the things that you get with more, a little bit more of a a glycosaminoglycan-rich section of the tissue. So what we found is how this was affected by age. So just wanted to make a note here that in general, the radially oriented test strips had a greater radius of transition curvature compared to circumferential strips. And it was greater for the mitral valve free edge, which is very rich in glycosaminoglycans. And it did tend to increase with age, particularly in the free edge. So now, how is the valve changing with respect to age? Well, if we looked at the suckling pigs versus the kind of more young adult pigs versus the much older pigs, we find that if this, in this little free edge domain here, you can see a lot of blue for the Smovat staining. This is very rich in glycosaminoglycans, and it becomes progressively more yellow with more collagen infiltration over time. And so that's the free edge, which is normally very rich in glycosaminoglycans. It becomes more collagenous. That is one of the reasons why it is becoming stiffer. But then the center region of the anterior leaflet, which is normally more collagen rich to begin with, becomes dramatically bigger and thicker. And we found that instead of having kind of a really uniform amount of collagen, there was more marbling with glycosaminoglycans over time. Now, we also looked at a much earlier stage, so we did look at fetal development. You do find that this layered structure does appear very early in development. We were specifically looking at glycosaminoglycans, and we can find some just in the first, in the second trimester, actually, you can see some a little bit of layering structure here for versican staining in the second trimester. The first trimester, you can start to see some elastin fibers that are distinctively present in certain locations. And there is a distinctive change between the prenatal period and the postnatal period, this perinatal change with respect to at least proteoglycan deposition, which is what we've been interested in for a long time. And so this is due to the fetal shunts that are closing at birth, and so the mechanical loading on the left side of the heart begins to predominate at this stage. And so with respect to, let's see, let me make the point here. There we go. We looked for differences between the aortic valve and the mitral valve and how they changed with respect to proteoglycan composition during this transition period. And the aortic valve had a dramatic drop in staining for the proteoglycans, proteoglycan biglycan, which is strongly associated with collagen fibrillogenesis. And hair is the hyaluronan receptor for endocytosis. So both sulfated proteoglycans and unsulfated hyaluronan had differences in how they were being made and turned over, with a big change in the aortic valve and a smaller change in the mitral valve. And we thought that was interesting because at birth, there's a bigger change for the hemodynamics for the aortic valve compared to the mitral valve. So there's some mechanically driven matrix remodeling that happens just at birth. Okay, and we looked at this stage because we were particularly interested in congenital heart defects. But you can also think about the way that these changes with age influence what you see in extracellular matrix and cell phenotypic remodeling. So with age here going from the young adult, which is, here we go, from the young adult pig to the much older pig, you see a lot more collagen type 3 staining, very, very pronounced. Collagen type 3 is more of a networked collagen compared to collagen type 1. We wondered if its presence in the older valves was related more to playing a role with stress relaxation. We also find there's more staining for non-muscle myosin, which is a myofibroblast marker for valvular interstitial cells in the much older valves. And again, just tried to think about how we could link that with tissue remodeling. My lab studies a lot of glycosaminoglycans, and one of the things that we noticed was not only were there more or changes in the glycosaminoglycans that we could stain for, but we could also quantify the very specific types of glycosaminoglycans over time. And so we found that with age, there was a pronounced increase in hyaluronan staining in the free edge of the mitral valves, or not staining, that we could measure biochemically. And in the center of the anterior leaflet as well, there was an increase in a lot of hyaluronan staining, hyaluronan quantification compared to normal. Okay, so then next I wanted to talk about how these effects of aging influence valve cells. So there's a lot of extracellular matrix changes, a lot of stiffness changes, and of course valve function overall is different with respect to age. How do these affect the VIC and VEC functions, VECs being the endothelial cells? So one of the first studies that we did on this is we looked at the cells from the mitral valve, and again alluding to the beautiful heterogeneity that has been mentioned today. We looked at this a little bit more simply. We harvested cells from the center of the anterior leaflet. So this is a tissue that is largely exposed to tension, and then the cells from the free edge of the posterior leaflet. So these are tissues that will normally experience compression. So cells that normally experience very different types of loading. And these were cultured atop soft hydrogels that had 2 different levels of stiffness. There was here we go. So a 5% PEG hydrogel is about like 35 kilopascals of stiffness, and then about 350 kilopascals of stiffness for the 15% PEG hydrogel. And then we had cells from 3 different age groups, the young adult, and older. What we found is that there was no one particular response to increased stiffness. So what we have here is the white bars are the softer hydrogels, the black bars are the stiffer hydrogels, and then we have 3 different age groups and cells that normally experience tension and cells that normally experience compression. So again, the MVAC is what experiences tension, and the PML is what experiences compression. So the cells definitely responded to the change in stiffness. And so here for this 6-week group, there was a decrease in cell activation demonstrated by smooth muscle alpha actin staining. But that was the case for just the cells from the center of the anterior leaflet, not from the posterior leaflet. But then we had the flipped response in the 6-month time frame, and then not really a significant response at all for the 6-year-old time frame. So it really depends. We also looked at 2 different factors involved in collagen synthesis. Both prolyl hydroxylase involved in the earliest stages of collagen synthesis, and heat shock protein 47, a molecular chaperone. And we did find some significant differences with respect to increasing stiffness, but they weren't necessarily uniform across the valves. So here they were nice and consistent with respect to the 2 sections of the valve leaflet. But then we had, at the much older age group, we actually had a different response with an increase in HSP47 with respect to increased stiffness in the posterior mitral leaflet, the posterior free edge. So really there's not one response to increased stiffness across the valve leaflet. The cells can respond differently depending on the extracellular matrix that they're normally surrounded with, and the type of loading that they normally experience. There's just we need sort of a map of VIC behavior that not only reflects the kind of the beautiful symphony of extracellular matrix that surrounds them, but also kind of a dynamic appreciation of their responsiveness based on age and where they're located in and what kind of mechanics they're normally exposed to. And so the last thing I wanted to mention was how valvular endothelial cell function is affected by age. So there are many different functions of the valvular endothelial cells, or VECs. One that we were particularly interested in looking at was the hemostatic balance, the antithrombotic and thrombotic protein balance that means that we don't have a lot of clots forming on our blood valve, our heart valves. This is a pretty understudied aspect of valve endothelial biology, but it was one that I was interested in through a collaboration with an expert on von Willebrand factor. And so we wanted to see how this hemostatic balance would be intersected with age-related changes. And so for this study, what we did is we looked at gene expression and protein staining for a variety of antithrombotic and thrombotic-related proteins. So for the thrombotic proteins, that's where von Willebrand factor comes in. We also look at tissue factor and PI1. And then the antithrombotic proteins were ADAMTS13, which cleaves von Willebrand factor, tissue factor plasminogen inhibitor, and tissue plasminogen activator. So we found that there were a lot of very specific age-dependent changes in gene expression for these different factors. So if you try to think of hemostatic balance as a balance between the thrombotic and antithrombotic proteins, these ratios were definitely changing with respect to age. And then when we did the staining, we also found some changes with respect to age. And in particular, I want to draw your attention to the von Willebrand factor. So even though the gene expression of von Willebrand factor decreases with respect to age, we found tremendous increase in staining for von Willebrand factor within these heart valves. And you may have noticed this as well. If you stain for von Willebrand factor, sometimes you see staining for von Willebrand factor, which is secreted by endothelial cells and should just float away into the blood. You may see it actually permeated into the tissue. And with age, there's a lot of von Willebrand factor in heart valves. You know, what is it doing there, and what does it lead to? So one of the things that we looked at was, how does von Willebrand factor, how is it, what could it be doing for the valvular interstitial cells? And so here we just looked at the secreted von Willebrand factor, and we find that VECs from all age groups will produce von Willebrand factor, even though the amount is a little bit less with age. And when we looked at the fragmented von Willebrand factor, this is the VWF 140, we find that ADAMTS13 is active in all of these age groups, and it is cleaving the von Willebrand factor. It's actually cleaving, there's more fragmented von Willebrand factor with age. So we took the cultured medium from these VECs and applied it to VIX and looked to see how it drove calcification. And the production of von Willebrand factor is stimulated by histamine, so we controlled for histamine, but we found that in general there was a pronounced amount of calcification in these, in all of these different age groups when we treated them with the cultured medium, conditioned medium containing von Willebrand factor. So the VEC secreted von Willebrand factor drives calcific nodule formation by VIX. And so I also want to point out, even as pronounced as this was, that is just the secreted von Willebrand factor. What is the effect of the von Willebrand factor that is within these aged heart valves? And so I don't have an answer for you on that. That's something that we've been working on for a while. But I do want to wrap up and say that there is a real spectrum of cell and tissue function across age and, of course, across the anatomy, as you have heard from many other speakers today. You can do stiffness, substrate stiffness studies, and I think that's a beautiful area of work that a lot of people are looking into. It will affect cell activation. It will affect many different characteristics of their cell phenotype, but that matter depends on the age. It also depends on the region of the tissue that you harvest the cells from. And there are a lot of changes that can affect valvular endothelial cells with respect to age, and that can affect a lot of endothelial cell function. We in particular looked at the hemostatic balance and found that that can contribute to the aortic valve cell calcification. So just in summary, if you study valves from just one common, easily obtained age group or one valve region, you may be limiting your understanding of what could be a very complex behavior. And it may not necessarily represent the human ages of valve disease, and of course, since that is something that happens at a lot of different ages, that should make you think about what age of animal model you're studying. And so keep in mind the mechanical function of the heart valve changes with age and of the cell microenvironment. And that should be considered when you're addressing the design of tissue-engineered heart valves. So I'd like to thank my students who have contributed to this work, especially Libby and Lizelle years ago, Desi, Madeline, and Chris recently, and Eric and Eze right now. Thank you so much.
Questions? I think we have a couple over on this side. While they're chasing the microphone, I want to ask one burning one, if that's okay. Have you looked at all at male-female differences and perhaps even female differences, say, pre-pregnancy, pregnancy, postpartum, anything like that, how that plays out?
So I love the pregnancy studies. I think they're really fascinating. I can't comment on that. I know that Sarah Wells and her student Megan Martin have looked at that recently. As far as male to female, we haven't looked at that with respect to age, but yeah, we do find that the shear responsiveness is very different. So with mechanical stimulation these days, we're mostly looking at shear stress activation, and it was surprising to us how different the male and female VECs behave with respect to shear stress. We're hoping to present that in London.
Ganyar Podja, I'm a cardiologist from the Netherlands. Thank you for your overview. I was wondering if are you also doing studies about other cardiovascular risk factors, if that influences also the calcification of the valves, beside the aging of the valves?
Right. So definitely just female versus male gender is one of those. Other cardiovascular risk factors we've looked at have been just the presence of lipids. And then also the hyperglycemia have been the things that my group has been focusing on. Yeah, nothing to share with you quite yet, but I definitely think that those are factors that are also aging associated.
Thank you for the nice presentation. I am a cardiac surgeon and I do peds and adults. And I'm wondering if applying those stiffness, extensibility to repaired valves in peds and adults, I think we can provide samples, very, very small amounts of tissue to predict the outcome, because one of the most challenging points of repair is how long is this repair going to last? And is there predictors that can guide the decision toward the early replacement and avoiding extra surgeries for a ped or an adult? Thank you.
Thank you. I think that this whole question of predictive nature and how can there be a way of getting a sense of the nature of the disease and the progression of the disease and the success of surgical repair. One of the things that has been just so exciting to think about for the last several years is how induced pluripotent stem cells can provide a surrogate indication of these questions. And that is also something I'm hoping that we can comment on later on when we are in London for our next meeting, with respect to the behavior of cells from patients with Turner syndrome. So I think that what you are commenting on is motivating a lot of research, and I'll be very excited to see how that comes out. Thanks.
One last question.
Thank you, Jane, for your talk. So your last conclusion comment was about we should consider this in tissue engineering, right? That we have changing mechanical properties with age. I have two small questions. Does this mean we need to have age-specific implants, or will the body adapt and change it? And with that, maybe we can look at a Ross procedure where you can transfer a sometimes stiffer aortic valve to the pulmonary position, or the other way around, and it's the other way around, my apologies. But then we see remodeling and we don't see problems there. So how important is this finding?
Yeah, I think that with respect to the Ross procedure, I think that we can definitely learn a lot from that. I will have to pivot just a little bit to answer a slightly different question, which is just a little bit more about age-specific heart valves. I would think that one of the questions that I've always, used as a motivating factor for a lot of work that we have done is how valves in younger children have a different mix of cells than in older adults who might also be good targets for tissue engineered valves. And in particular, the more vascularized nature of the younger valves has been something that I feel like I haven't seen enough about, and I've always been trying to think about how the valves in the younger children could be designed for more growth and remodeling in a vascular-driven method compared to those that are implanted in adults. So those are just my thoughts on it, which I had to realize doesn't answer your question completely, but hopefully will spark some discussion. Thank you.
All right, let's thank all of our speakers again. Thank you for a fantastic session. We now have time for a coffee break, so I'm going to take a 15-minute coffee break and ask that everyone be back here at 11:15 for the next session. Thank you.